All right, let's review for this first exam. So, in doing so, the things that I want to uh, highlight here are both what you can expect and what you cannot expect. So, in terms of things you can expect, my exams tend to be big picture conceptual exams more than trivial facts, I suppose. I'm not going to ask you to recall minutia off slides or out of the book. That is not helpful. What you should be focused on are big picture ideas where things tie together in such a way that it creates sort of a unified theme. Because that's really what we're going for here, not just remembering stuff for the sake of remembering it, but applying that in some way. Because that's the point of all of this is you to be able to apply things. So, what should you be focused on? Well, our biggest topics thus far have been uh, cells and histology. And I suppose the skin to some degree. Things I'm not worried about. Don't concern yourselves with the history of anatomy. That's obviously not a topic of real relevance for you. It was more of an icebreaker for us. Um, in fact, let's just go sort of in chronological order here in terms of topics that we've talked about and what you can expect. So, the first topic of any real importance to you would be directional terms. So make sure that you are able to apply those anatomical directional terms. Again, I don't expect you to memorize them and just regurgitate that. That's not helpful. But you should be able to, for instance, tell me that the head's superior to the neck, or the skin's superficial to the muscle, or the heart's medial to the lungs. This sort of thing. This is useful. But again, I'm not going to ask you like definitions of those terms. I'm not going to ask you to identify quadrants. Um, be able to recognize the planes. So your sagittal and your transverse and your frontal planes, be able to recognize those because you will see that in terms of like what kind of plane is this, and you should be able to identify that as a transverse plane if you see that section on the exam. So there's that. Also, um, understand the process of negative feedback. When the product of a system inhibits the system. This is going to come up repeatedly through the semester, so you do need to understand what that means. Okay, I'm not terribly worried about chemistry because most of our chemistry stuff is going to be sort of discovered through application, where um, you'll catch on to this because we use it a lot. Things you should be familiar with, however, we have our organic molecules, our carbohydrates, proteins, nucleic acids. These are four classes of organic molecules. Know their basic function. I'm not going to ask you to identify a carbohydrate. I'm not going to throw one out there and say, is that glucose? There's very little help, like in terms of your clinical career, that's going to rely on you being able to identify glucose and galactose and fructose. But you should understand that carbohydrates are typically fuel for the cells. Lipids, Proteins. This is all important. Proteins. We spend a lot of time with proteins. Specifically, you should know that an enzyme is a biological catalyst. It's a protein. And like all proteins, we have different levels of structure. So, primary, secondary, tertiary levels of protein structure. Make sure that you associate that tertiary level of protein structure with the three-dimensional shape of the protein. This is what's going to determine function. The nucleic acids, right? DNA, RNA, genetic material. You do need to know that ATP is our energy molecule. Let's see. Sorry if I pause here, it's because I'm reading through your exam to make sure that I'm covering what I want to in this review. So, on that note, as we talk about this, keep in mind that the subjects that I discuss during any sort of review like this are coming straight off the exam that I've written. Meaning that if you have to focus on something for your studying, this is the guide to focus so that you're not trying to spread yourself too thin here. Focus on what I'm reviewing because I'm literally going through your exam as we do this to make sure that I'm covering topics that I've asked you questions about on the exam. Okay, so we're going to move up to cells. Again, the chemistry that's important here is the chemistry that's important to the cell. So I'm not going to ask you to balance equations or talk about chemical bonding. You need to know that lipids are nonpolar, so that makes them hydrophobic. I'd also like to take this moment to apologize for my writing. It's much more difficult to write on a computer screen than one would expect. And, all right, lipids, nonpolar, hydrophobic. Our other things like proteins and carbohydrates and nucleic acids are molecules, which makes them hydrophilic. So, this comes up in terms of how the cell works. So, membrane transport is a big topic here. Things that you need to know with membrane transport. All right. Remember that your cell membrane is a phospholipid bilayer. So that means that we get our structure that looks like this. phosphate heads, fatty acid tails. That is polar. Because of the way the cell membrane then is structured, that means that it is freely permeable. Now this is a big thing here. Sorry, I'm looking for the highlighter thing, but it's just as easy to circle as. Lipids freely cross the cell membrane. So lipids freely cross the membrane. What are lipids that we talked about? Fatty acids, steroids, fatty acids. If it's a lipid, it will freely cross the cell membrane. You will see questions on this exam, and I'm telling you this now. You're going to see questions on this exam that say, which of these molecules can freely cross the cell membrane? Pick the lipid. So, for instance, things that could hypothetically freely cross the cell membrane. Any steroid hormone. Has to freely cross the cell membrane. Things that can't. Anything that's not a lipid. So if you have a list of things on there, and some of them are lipids and some of them are not, and it says which of these cannot cross the cell membrane freely, pick for instance, the protein. Lipids freely cross the cell membrane, and there's at least 2 questions on this exam that just rely on your ability to know that lipids freely cross the cell membrane. Oh, okay, here we go. Let's put this together. So I'm going to kind of do this whole thing where we're putting these big ideas together. Okay, so we've got this. Lipids freely cross the membrane. Now, remember what we have for passive transport. So we have diffusion. Simple diffusion is limited to lipids and things that are lipid soluble. And then we have facilitated diffusion. Facilitated diffusion, remember, is when a protein acts as like a bridge so that we can cross the cell. This is for non-lipid soluble, so polar molecules like carbohydrates and stuff have to go through facilitated diffusion, or charged ions. Like we're going to have to use a protein to cross through the cell membrane as like a bridge or a gate, or even like a channel of some sort, so that things that are not lipid soluble can go inside and outside the cell. Remember that with passive transport, we're always going from a high to low concentration. And then, of course, we have osmosis, which is the diffusion of water. Remember your rule with osmosis. Water always moves to the area of higher concentration. So first, let's take a look at this idea of osmosis. All right, so cell, outside the cell, right? So water always goes to the area of higher concentration. If I put a bunch of stuff out here, outside the cell, this is hypertonic. In a hypertonic environment, there's more stuff on the outside of the cell than there is on the inside of the cell, so the water goes out. In a hypertonic environment, water leaves the cell. Hypertonic. When you think about hypertonic, it's like putting salt on a snail. What you do there is you make the outside of the snail hypertonic, and all the water leaves, and the snail dies horribly. That's for science. It's fine. Okay, so hypertonic. Water's leaving the cell. All right. So what then if we put the stuff inside the cell, or if there's more stuff inside the cell than on the outside of the cell? Well, now we're in a hypotonic environment. In a hypotonic environment, the concentration is higher on the inside of the cells, so now water goes in. Neither of those is ideal. If the water's going in the cell, it's going to freaking explode. If the water's going out of the cell, it's going to shrivel up and die. So your cells are in an isotonic environment, where the concentration is the same on the inside and the outside. So now water moves both ways, but in equal volumes. So we don't get a net gain or a net loss of the cell volume. Tonicity is another important topic for you. So make sure that you go back and you review tonicity so that you can tell me if the water's going in or out of the cell. So if I tell you, you put this red blood cell in a hypertonic environment, where does the water go? In a hypertonic environment, the water leaves the cell. You put the cell in a hypotonic environment, the water is going to come into the cell. Now, in the interest of making this palatable in terms of review, I'm going to break it up into segments. So this will be the end of the first segment, and then we're going to immediately start the second segment and kind of pick up right here. So part one, I suppose,